Hello, this is Melissa Davis at the GRU UGA Medical Partnership, and this is an introduction to cancer genetics. Listed here are the learning objectives, which includes explaining the concept of how multiple mutations in distinct genes and gene types cause sporadic mutation. We'll apply the two-hit hypothesis or theory to specific forms of familial or inherited cancer. We'll also characterize cancer genes into the categories of proto-oncogenes and tumor suppressor genes, as well as discuss the role of DNA repair mechanisms and the general characteristic of genome instability in cancer. Lastly, we will investigate other causes of tumor etiology, including epigenetic regulation and the impact of environmental agents. Two unifying things about cancer genetics is first that cancer is a disease of the genome, and secondly, cancer has a different inheritance pattern than that of other genetic disorders. Concerning the disease of genes, basically, multiple cancer phenotypes arise from the fact that there are mutations in genes that regulate cell growth and division. These mutations usually arise by environmental exposures to chemicals that increase mutation rates. We call these chemicals mutagens. Some cancers are caused by viruses that carry genes that promote cancer. And then also we see in certain cancers unique chromosomal changes, including structural changes and changes in numbers of chromosomes. Lastly, in terms of inheritance patterns, one can inherit a predisposition to more than 50 forms of cancer that we consider to be inherited or familial. These are usually referred to as cancer syndromes. Also, inherited mutations usually predispose someone to cancer, but the mutations that actually cause the tumor occur in the somatic cells. And it is the mutations in these somatic cells that accumulate in clonal descendants of a single cell. So what is cancer? Most define as simply uncontrollable cell growth. Well, there are some very basic characteristics that we see on the cellular level that help us to determine the difference between normal cells and cancer cells. The first is that usually in normal cells we see a large amount of cytoplasm surrounding the nucleus, and usually we see a single nucleus and a single nucleolus. In these cells, the chromatin is very fine, and oftentimes you can't see any defined structure in the nucleus, especially during interphasic cells. However, in cancer cells, we usually see sometimes multiple nuclei, multiple nucleoli, and then also the nucleus itself seems to enlarge and takes up the large proportion of the cell structure. These images to the left and right are immunohistochemistry images that basically show us the cell structure of a normal breast duct and then what a malignant breast duct looks like. One other characteristic of cancer cells is the fact that they resemble stem cells. We believe that there's a process called dedifferentiation in which cells that have been fated to a certain organ, so they have a specific cell type, go through a process of losing this cell type characterization and resembling something that looks as though it is a stem cell and has the ability to become different other types of cells. This process of dedifferentiation is actually what allows the cell to have no cell division limit, and this is how uncontrolled cell growth essentially develops. So let's talk about cancers in the US. Most common cancers are listed here, and of course the most common cancer in males is prostate cancer, and the most common cancer in females is breast cancer. If you click on any one of these links, it will take you to the NCI and the CDC website that basically gives us the recent statistics concerning the most common cancers in the US. When we get to class, we're also going to visit the NCI website that lists state-specific statistics. One of the things we pay attention to in terms of the onset of cancer is, of course, age. So age is a leading risk factor for cancer. Essentially, the longer you live, the more likely you are to develop cancer, and this is usually sporadic. Okay, so sporadic cancers usually occur later in life. Cancers that occur earlier in life are usually the result of some inherited predisposition or cancer syndrome. So, although we hear a lot about inherited cancer syndromes and inheriting a predisposition due to mutations in certain types of genes, the far majority of most cancers are sporadic. These are caused by an accumulation of a number of mutations in somatic cells, and we'll go through what those gene types are later. But what I would like to point out is that only a single cell needs to acquire this superpower to become malignant. Okay? In inherited syndromes, this predisposition doesn't always have to result in tumorigenesis. While the first mutations are carried in all cells but in a heterozygotic state, usually in the two-hit hypothesis model, there needs to be a loss of heterozygosity where you actually lose the function of the normal allele that you've also inherited. One reason that breast cancer, for instance, does not follow a very distinct inheritance pattern is usually because of incomplete penetrance. This simply means that you can carry the mutation that predisposes you to this cancer without ever actually getting cancer. So what are the types of cancer genes? Largely, we can divide cancer genes into two broad categories, oncogenes and tumor suppressor genes. Both of these categories have in common the fact that these genes usually regulate some function of the cell cycle. Okay, so proto-oncogenes are usually the genes that initiate or maintain cell division that become cancer genes when they acquire a mutation. Okay, oncogenes usually act in a dominant manner, and they're usually a part of some signaling pathway. So basically, we've lost the ability to turn these genes off. Okay, in tumor suppressor genes, these genes usually suppress cell division. Okay, so a lot of these act as checkpoints in the transition from one phase to the next. And usually, when we delete or inactivate these gene products, that allows the cells to continuously divide. So essentially, these cannot act dominantly in a predisposition or heterozygotic state because we still have the good copy of the tumor suppressor gene. Although in reduced function, it's still present, and usually just having one copy is enough to maintain regular cell division. When we think about dominance relationships between oncogenes and tumor suppressor genes, we usually consider that the dominant oncogene is such because when you mutate an oncogene or a proto-oncogene and it becomes an oncogene, usually the result is that there's an abnormal protein or there's an excessive amount of the protein. So again, you have overexpression of the gene, such that this overexpression overcomes then the normal expression regulation of the normal allele. All right. In a tumor suppressor gene situation, usually the mutated gene results in an inactivated protein or removal of expression of that gene. But as long as we have a normal copy that produces the normal protein, we can still have normal cell proliferation. However, once we lose both copies of the gene, this is when we start to see excessive cell proliferation. So, with one mutated gene copy, you still have a normal protein. Therefore, in a heterozygotic state, you still do not see the manifestation of a tumor forming. This is by definition recessive. We also categorize these as gain-of-function mutations. Okay, we discussed this in the first week, where we've either started expressing the gene in areas that is normally not expressed, or we're expressing more of the protein, or we have hyperactivated the protein in some way. Loss of function mutations are usually the result of tumor suppressor gene mutations. In this case, it's very clear we've lost the function of that tumor suppressor to regulate cell division. In the two-hit model, basically what we're saying here is that once the first mutation arises, this establishes a predisposition. Okay, you can actually inherit this first mutation, or you can acquire it. However, it takes at least one more mutation, either in that same gene or in another gene with similar function, before you can reach the state of cancer formation. You can actually carry this first mutation and still have controlled cell growth, so no tumors forming, until you acquire the second mutation. The number of hits required to reach a tumor depends on the specific genes and the function of those genes. A classic example of genetic predisposition to certain cancers is the retinoblastoma mutation. Retinoblastoma is usually caused by mutations in the RB or retinoblastoma gene. We know that retinoblastoma functions in eye development, and individuals who inherit one copy of the RB negative or RB null allele are prone to have cancer in the retina. Somehow, during proliferation of retina cells, the RB allele is lost or mutated. This is what's called loss of heterozygosity. And then tumors actually develop as a clone of those cells that have lost the allele. So here in the schematic, basically we show that this young man has inherited an RB null allele from mom, but a normal copy from dad. So he's in the heterozygotic cancer-prone state. During eye development, some of the cells of the retina can lose, one of the good copies of the RB gene. This results in overproliferation of that cell, and such that the tumor is actually a clone or a colony of clones of that one cell that lost the RB gene. I want to take this opportunity to also talk about the fact that this tumor suppressor gene has a specific function in regulating the G1 to S transition in the cell cycle. It is a transcription factor, and it regulates the activity of an important protein called the E2F protein. Without RB, we also see other genomic instabilities. So this is a karyotype from a retinoblastoma gene, or retinoblastoma tumor, And what we see here is a lot of chromosomal abnormalities. We see fusions of different chromosomes. We also see isometric chromosomes where the centromere is lost. Another type of proto-oncogene is particularly cell signaling is the RAS oncogene. Normally, RAS is inactive until it becomes activated by the binding of specific growth factors. This binding of growth factors usually causes a GDP to GTP transition, and then proliferation occurs. In cases where we have mutations in RAS, the GTP is constant, and so you don't actually need binding of the ligand or the growth factor to initiate proliferation. This is a gain-of-function mutation. Now let's talk about some of the specifics surrounding genomic instability. Another category of mutant cancer gene affects DNA repair, and this directly then contributes to genome instability. Some of the basic properties of cancer result from the inability of cancer cells to be able to repair the damage to their DNA. This then results in high rates of mutation, chromosomal aneuploidies, and genomic deletions and duplications. These DNA repair genes, as they are called, are now recognized as a specific class of cancer genes, right along with tumor suppressors and proto-oncogenes. Two classic examples of DNA repair genes are the BRCA1 and BRCA2 genes. Mutations in these genes are what predispose women to breast and ovarian cancer, respectively. Specifically, we now know that BRCA is usually only found in the nucleus, which gives us a clue to its function dealing with DNA. It stops DNA replication as it binds to this protein called RAP80. The RAP80 protein identifies DNA damage and initiates its repair. Therefore, in the absence of BRCA1, RAP80 never gets to recognize DNA damage, and repair is never initiated. In this way, we tend to see within cancer cells where there's a DNA repair mechanism disabled, a large amount of mutations. Okay, so mutations normally occur during regular DNA replication, but we have this machinery, the DNA repair machinery, that basically finds it and corrects the mistakes. Okay, if the mismatch is not corrected, then you see the mutation fixed in the DNA sequence. When we say increased mutation rates, basically what we're saying is that we see an increase in these types of mutations that occur normally during replication. If you couple this with environmental exposures to mutagens, people who have a reduced functionality of DNA repair need to protect their DNA, as I say, by limiting their exposures to certain mutagens. Another type of genome instability has to do with chromosomal structure. Okay, so this is a image of a spectral karyotype where basically we use different dyes or different probes that paint each chromosome a unique color. We can also use probes to create a certain spectrum across all chromosomes. This allows us to be able to visualize translocation events more readily than the normal G banding patterns and karyotypes. Some rearrangements we know are very unique and specific to certain tumors, such as the Philadelphia chromosome and CML, and we'll talk about that a little later. Now on to the molecular genetics of cancer. One of the things I really want you guys to take home is the use of genetic tests for cancer diagnosis and prognosis. So while some of these technologies are still considered emerging in the clinic, depending on where you end up practicing, some of these are very commonplace. For instance, fluorescence in situ hybridization or FISH, IHC or immunohistochemistry, where we can also use tissue microarrays, and I'll tell you what those are in class, and DNA expression microarrays, which basically test the expression levels of all of the genes. We use FISH analysis, for instance, to diagnose subtypes of breast cancer. The HER2 amplification is literally a manifestation of multiple copies of the HER2 gene. So in FISH analysis, we basically have a probe that recognizes the sequence of the HER2 gene. And in normal cells, we only expect to see two little blobs. In this particular image, the green probes are the control probes, and usually our control probes represent the location where we expect to see the HER2 gene. All of these other red probes then indicate severe amplification of the HER2 gene itself. This then results in the overexpression of the HER2 gene. We can also use immunohistochemical analysis for certain genes, for instance, for estrogen receptor status. So in this particular IHC from my lab, we have brown staining that indicates the expression of the estrogen receptor. So this particular invasive tumor is ER positive. For expression profiling, usually we use this only in experimental capacities, but certain clinical trials are now relying on the profiling of a suite or a subset of different genes. In order to look at multiple genes from a single sample effectively and efficiently, we need to use a method that will allow us to interrogate all of the genes at the same time. And so basically, a DNA microarray analysis will allow us to do that. So given a subset of genes, and usually these genes will be in a particular signaling pathway, if we see that subset upregulated or downregulated, that allows us to classify a certain case in terms of favorable or unfavorable. And we'll discuss some of these gene reports in class. Now, as far as the Philadelphia chromosome is concerned, this is a very unique translocation that occurs in CML very often. What this indicates to us is that it is possible to cause a chromosomal rearrangement that activates an oncogene. In this particular case of the Philadelphia chromosome, we have a reciprocal translocation between chromosome 9 and chromosome 22 that occurs in a break that fuses together the ABL gene and the BCR gene. The fusion of these two genes creates a new gene, which also becomes a new drug target. So essentially what we've done in translocating these two chromosomes, especially at this break point, and usually you'll see this report on the gene report T922, and we'll talk more about this nomenclature later in chromosomal aberrations. And it creates a hybrid gene, which actually gets translated into a hybrid protein. It is this novel hybrid protein that yields uncontrolled cell growth that causes CML. Because of its uniqueness, we can actually target this hybrid protein with targeted therapies to reverse or cure the CML. Another indication of genome instability that we can use with either FISH for specific probes or just a general DNA stain is something called double minutes. Okay, so normally we expect all of our DNA to be associated with specific chromosomes in the nucleus. However, when we lose the ability for DNA repair, we not only lose mismatch repair, which I discussed a couple of slides back, but we also lose breakpoint or chromosomal break repair. When we lose chromosomal break repair, we start seeing the accumulation of fractions of chromosomes, and we call these double minutes. They actually undergo DNA synthesis as though they were still part of a chromosome. Another indication of genome instability is something we call microsatellite instability. Microsatellites are basically DNA repeats that we find throughout the genome on just about every chromosome. Usually, the size or length of these repeats is maintained at a certain molecular size or nucleotide size. When we have microsatellite instability, we start seeing an increase or larger microsatellites, and we also see more of them in the genome. So this increased size variability creates a shift in the average length, which we are able to see using capillary electrophoresis and other mechanisms such as that we talked about in week 1 and that were discussed in the medical genetics spa last week. The genes that are usually responsible for microsatellite maintenance are MLH and MSH genes. Oftentimes, we'll see mutations in these genes in colorectal cancer. Now let's discuss the multi-step mutation model. There was a large collaborative effort called the Cancer Genome Atlas, where essentially hundreds of labs around the world decided to sequence the genomes of tumors that were collected from different cancer sites. The premise here was that as we catalog all of the mutations that have occurred in cancer cells, we can eventually start paring down which mutations actually cause the malignancy versus mutations that accumulated along with the instability of DNA repair, and they're therefore called passenger mutations. What we see accumulating are what we would expect to see, right? So we see, for instance, RB overrepresented in multiple sites, not just, of course, eye cancer. We also see other cell cycle regulation genes overrepresented in multiple sites. But what's most important is that we see multiple mutations in every type of tumor. This is what is thought to be the accumulation theory. In sporadic cancer, which occurs again later in life, we believe that there are successive mutations in successive clones that cause the proliferation of malignant tumors. So we start with mutation 1, which may give some selective growth potential. Then we acquire mutation 2 that also now gives an additional growth potential. Then we have mutation 3, and so within each subset we start to see a new colony of clones that have accumulated different mutations and are passing those on to the daughter cells. Eventually we come to a mutation that gives that cell a superpower to be malignant. So it has now evolved and evaded all of the body's checkpoints, all of the molecular checkpoints to help keep overproliferation in check. One key example of the multistep mutation model is in FAP-associated colon cancer. Usually the first mutation is in the APC gene. The second mutation is in the KRAS gene, which we talked about earlier. Then we have mutations in the DCC gene, which by this point we see late adenoma with villi, which indicates invasiveness. By the time we get to metastatic cancer, there could be multiple mutations, including those in P53, which is a well-known oncogene, tumor suppressor. What we recognize from the Cancer Genome Atlas project is that the accumulation of these mutations often means that we have lost the ability to maintain very specific pathways. Or, as I like to say, the cell has acquired multiple superpowers. So in combination, many of these categories, once you acquire mutations in genes that regulate these categories, can lead to metastatic carcinoma. So some of these superpowers are the ability to invade, so remove itself from its original site. Of course, it must obtain limitless replicative potential. We also see in more aggressive types of tumors sustained angiogenesis, where it is recruiting blood vessels into the tumor in order to feed itself. It evades apoptosis, which is basically the cell death pathway. And it also becomes self-sufficient, meaning it doesn't necessarily need growth signals, for instance, in the case of the RAS mutations. And lastly, it's become insensitive to anti-growth signals. And usually these are the result of the immune system. These last several slides are just here for your information and to serve as a study aid for you. We will talk about many of these topics and specific types of cancer once you get to class. Thank you for your attention.